Great. Thank you very much for inviting us to speak here. I'm Laurent Fischer. I'm the CEO of Adverum. We're an ocular gene therapy company based in the Bay Area in Redwood City. Those are more forward-looking statements. We're talking about gene therapy here for a very large population. There's, in fact, 1 out of 200 people over the age of 60 and 1 out of 5 over the age of 90 that develops age-related macular degeneration. About half of it is dry AMD. The other half is wet AMD. Wet AMD is actually treated by injections in the eye every few weeks. It's a lifelong commitment and is a real treatment burden, and as well as a very significant impact on healthcare resources. In fact, about 13% of Medicare Part B in the US is spent on anti-VEGF for wet AMD. So the hope is that with gene therapy, truly regenerative medicine, we can have a one-time injection intravitreally and provide a lifelong benefit of vision preservation and significant cost effectiveness, as well as potentially cost saving for the systems. Ixo-vec is an aflibercept encoding AAV2.7m8 vector that uses a global promoter that is actually injected intravitreally. 7m8 refers to the loop on the capsid of AAV2 that crosses the inner limiting membrane, allows to transduce the back of the retina. It really is the gold standard as far as intravitreal gene therapy for ocular diseases. It's actually currently in 7 programs. 2 are in the clinic. Several others are about to go into the clinic. Has been licensed to multiple companies. And something that really has been one of the holy grail. It does not require a complex vitrectomy, subretinal surgery, which is applicable for rare disease, but not so much for large indications. So the current standard of care is actually these frequent injections of anti-VEGF in the eye. There's actually a bolus mode with a treatment extend mechanism. And they work well as far as restoring vision, but over time, vision gains are lost. And in fact, up to half of the patients stopped taking their injections over 2 to 5 years. So there's a real need to show actually stable levels of anti-VEGF, like aflibercept, which we can deliver with Ixo-vec. The hope is that we can really shift that curve and actually allow patients to maintain vision for the rest of their lives. In fact, even the newer anti-bolus treatment, Vabysmo, a bispecific antibody, and Eylea HD, while they can actually improve vision in treatment-naive patients, you can see on those graphs on the left-hand side, you can see that there's a trend over 2 years, even in the best setting of a clinical trial, to see a slight loss in vision. You can also see the bottom left-hand side, this actually fluctuation in the central subfield thickness, a marker of fluid accumulation in the retina and in the macula. And these actually changes in the macular thickness are known to lead to worse vision outcomes. So obviously, we have great treatments since the early 2000s for these diseases that had no treatments before that. Because of the treatment burden, there's been a lot of push to try to extend treatment interval. We've gone from every 4 to 8 weeks to 4 to 12 weeks, 16 weeks in some cases in some patients, and there are now treatments looking at 6 months. But the only way to really hope to be able to have lifelong vision benefit is with gene therapy. And in fact, there are 3 programs that use outpatient injection system like intravitreal or suprachoroidal that are in the clinic. But Ixo-vec, Adverum's proprietary vector, is the one that has the longest data with this method of administration past 3 years. So our first in-human study was called OPTIC. It was a study that looked at 2 doses of Ixo-vec, 6E11, 2E11, in patients that required frequent treatment injections on an annualized basis, about 10 injections per year, 1 every 6 weeks. And that study started over 5 years ago. We have patients who have been 5 years with a single injection with stable vision and dry maculas. This actually study looked at a very short prophylactic regimen of either 2 weeks of oral steroids or 6 weeks of steroid eye drops. And as you can tell here in these patients, we presented the 3-year data last year. We actually see very stable vision, dry maculas, and in fact, at 2 years and 3 years, more than half of the patients were entirely free of injections, with overall an 84% reduction in annualized anti-VEGF. Really a significant improvement upon the standard of care. And Ixo-vec was well tolerated with minimal anterior chamber cell inflammation. They were all manageable with steroid eye drops. Based on that, we actually started a phase 2 study where we actually decided to look at a lower dose of Ixo-vec. So we went from 6E11 to 11. Now we're looking at 2E11, 6E10. We also looked at an enhanced prophylactic regimen, tried to optimize this prophylaxis to minimize the risk of inflammation, which is all mild to moderate interior cells, non-symptomatic, but trying to really minimize any risk of long-term complications. So this study was fully enrolled last July. As I mentioned, we're looking at 2 doses in treatment-experienced patients, similar population to what we studied in OPTIC, for which we have long-term follow-up. And actually, patients were randomized to receive in a masked fashion, either 2E11 or 6E10, and they had a combination of local, with or without systemic steroids, to try to understand the contribution of old prednisone in the immune response you see in the eye, which, as we all know, is a partially immune-privileged organ, but not entirely, and we thought it was important to understand that contribution. The supplemental injection criteria for patients who are in the study are very standard, either an increase in CST by 75 microns or loss of 10 letters of vision or new vision-threatening hemorrhages. You can see here actually comparing on the right-hand side the OPTIC population to the LUNA population, very similarly high treatment burden, about 10 annualized anti-VEGF the year prior to coming to the study. Slight difference in a slightly better vision in the second study, LUNA, as you would expect. Patients who have slightly better vision coming to these first-in-human trials. And then slightly drier or better controlled fluid with CST that went from 397 to 350, and about a third of the patients being phakic, so having their natural lens, something that you cannot really do when you do subretinal gene therapy. So as you can see here, this was data we presented at the Macular Society in February. This is early result from the study. All patients had been to week 14, a fraction to week 26, which is going to be our next interim analysis coming out later this year. You can see roughly a 90 to 94% reductions in annualized anti-VEGF. That compares well to the 86% we saw in OPTIC at the same time points. So very significant reduction, confirming what we've seen, and that both doses seem to perform well. Looking at patients who are truly injection-free, you can see 68 to 85% in LUNA versus 73% in OPTIC, and that's actually, again, a very high number. If you look at the entire patient population in this slide, which is a swim lane plot, these patients are masked according to dose so that we cannot unmask the study. You can see 75% of the patient overall were free of injections at the different time points, and then we saw a 93% reduction in annualized anti-VEGF, and only 6 out of the entire population, so 10% required then more than 1 injection. So we think this is a significant improvement. You can see on the left-hand side all the multiple anti-VEGF that were used, and patients sometimes try to go from one to the next, to the greatest one, to see if there's an improvement in the treatment interval or in vision. And we'll get back to that in a second. So importantly, BCVA was maintained, and you can see that this was similar to what we saw in OPTIC, the dotted line in blue. BCVA was maintained out to week 26. We saw that out to 3 years, and we followed this patient out to 5 years in OPTIC. And CST, which was slightly higher in the OPTIC, it means there's a slightly larger drop in fluid at the last time we saw these patients. But in all 3 doses, you see a very nice drop in CST, and this is completely stable. So really a drying effect of the anti-VEGF, which you see sustained and stable, unlike the bolus injection that we use today. We actually looked at the aflibercept level in the anterior chambers. Now remember that the aflibercept is actually made in the retina, crosses the front of the eye, and we're trying to understand whether there is therapeutic threshold to find out what was the optimal dose. In fact, there's no real therapeutic threshold. We know that their patients are below the limit of detection of the assay that still has 0 injections because most of it is made at the site of the disease and probably controls VEGF quite well, so you probably need very small amounts. But the blue lines show the patient we followed out to 4.5 years, and we have completely stable levels of aflibercept in the eye for 4.5 years, and I would expect that as we continue for these patients, very much this is likely to be a lifelong benefit. The dots on the week 14 is when we actually took the sample from the LUNA trial, and actually you can see there's actually a nice therapeutic range with a nice overlap, which is something we had seen in non-human primates. So we believe those 2 doses are likely to be effective, and we'll obviously select 1 or 2 doses to move into phase 3 when we have our interaction with the regulatory agencies in the next months. We looked at we're trying to re-understand how can we optimize prophylaxis, because we know that steroid eye drops work, but they require compliance and the ability to hit your eyeballs every time you put a drop in your eye. If you do that, sometimes it's challenging. So we actually tried to use a Nosodect implant, a bioerodible implant of dexamethasone, to see if that would be sufficient. It turned out that it worked very well early on, but it does kind of drop pretty steeply as far as PK. After 6 to 8 weeks, we saw some breakthroughs, actually at week 8, 10, 12. So we added drops on top of it. You can see that we have very nice control with 3/4 of the patients being free of inflammation entirely, about 17% with traces cells, and 9% essentially with one cells. And we also saw there was no contribution of oral steroids, which is obviously something that we wanted to understand, somewhat of a relief, because in this elderly patient population, steroids are not always well tolerated, but we thought this was an important fact to understand. You can see here actually that in the dipyrifranate on top, the combination of Ozurdex and dipyrifranate on the bottom, sorry, the opposite, in yellow, the combination, in blue, the drops only. You see just traces of cells in the interior chambers on slit lamp exams, but these were all manageable with local steroids. And we see some transient, some pigmentary cells as well. We've seen also some TIDs in those patients. This is something that's seen commonly in ocular therapy, whether it's subretinal, suprachoroidal. There's some pigmentary changes seen with viral vectors, but they're not adverse and have no impact on vision. If you look at the anatomic changes in patients who have the highest amount of fluid, we're looking at actually over 400 micron at baseline, and comparing in blue, again, optic, to the 2 green colors for the LUNA trial, you can see that actually even patients with the highest amount of fluid see a very nice drop. That drop is completely sustained. That's not necessarily the case with all therapies that are in development today. We're very pleased to see that. And if you look at the patients that were on Vabysmo, the bispecific antibody that was approved a couple of years ago, you can see that some of those patients went back and forth across multiple different anti-VEGF. They all received that orange dot is actually aflibercept at baseline to make sure they respond to anti-VEGF before they receive a permanent aflibercept gene therapy. And you can see that all of them to date have been totally free of injections. So clearly, a very significant improvement upon today's standard of care. Comparing the various gene therapy programs that are so like either intravitreal or suprachoroidal, you can see that with Ixo-vec, we have significant improvement in both the reduction in annualized anti-VEGF as well as the percent of patients that are injection-free. What we also have is the long-term follow-up with more than half of the patients being free of injections and about 84% reduction in annualized anti-VEGF. So potentially a very significant benefit for all stakeholders, including payers. I mentioned the fact that fluctuation in the amount of fluid is actually believed to have this trampoline effect that leads to poor long-term vision outcomes. There are multiple papers that were published from Europe and other institutions. And it makes sense when you start looking at these case studies. This is actually a patient that came into the study, was well controlled, but you can see that patient on the bottom left, there's actually a spike when that patient missed a visit, which happens often when you're 60, 70, 80, 90 years old. You may be hospitalized, you may have a fall, and you can see how quickly the fluid accumulates. So there's really something about that treatment paradigm that's not really controlling the disease unless you really constantly attach to your retina, especially in getting those injections. You can see that that patient responds 2 weeks after the aflibercept. That patient has no fluid, but you can see now, past 3 years after single injections, the vision is actually improved by 3 lines of vision from baseline, and the retina is dry with CST being actually at physiological levels. So a very nice improvement. So we actually confirmed from LUNA to date what we've seen in OPTIC is that we have a good long-term safety, good disease control, and long-term durability. We believe we have a best-in-class efficacy and an acceptable safety profile with a go-forward regimen, and our plans to initiate phase 3 in the first half of 2025. We have received fast track as well as prime designation and ILAP in the UK, which allows for enhanced interaction. We will be hopefully looking for European partners at some point to advance this into phase 3. That's why we're here. And we have multiple programs that are in earlier stages. We have multiple collaborations. We actually spun out a program into BlueGen, and we licensed our capsids to a few partners that are actually, I believe, at this conference, Gencyte and Ray Therapeutics. So we really go focusing on the 6 months, 26-week interim analysis that will come essentially mid-year, and FDA and EMA interaction to harmonize the phase 3 program with the goal to start the phase 3 in the first half of next year. Thank you.